Right. Soluble mediators of the immune system, which are molecules released by immune or other cells that dissolve in a body fluid like blood or the tissue fluid and can help immune cells communicate and coordinate a response. These are your learning objectives. All right, so we have the complement system. So the complement system is a group of plasma proteins that can enhance innate immunity. Activation leads to pathogen opsonization, inflammation, and lysis. There are three main pathways of the complement activation. So the first one is your classic pathway. This is triggered by antigen-antibody complexes, and it connects adaptive immunity to the innate immune defense. It leads to activation of the complement proteins, resulting in pathogen destruction. The next one is the alternate pathway. This is triggered directly by pathogen surfaces without antibodies and provides a rapid innate defense against infections. And then the last one is the mannose binding lectin pathway. This is triggered by binding of mannose binding lectin to sugars on microbial surfaces, and it activates complement to enhance pathogen clearance. There are three main physiologic activities of the complement system. So we have the host defense against infections. This is the immune system that protects the body from microbial infections. It combines innate and adaptive mechanisms to detect, attack, and eliminate pathogens. And then we have an interface between innate and adaptive immunity. So innate immunity provides the first line defense and alerts adaptive immune cells. Adaptive immunity provides specific long-lasting responses, and this interface ensures coordinated and effective protection. Then we have disposal of waste, so the immune system helps remove dead cells, debris, and damaged tissues. Phagocytic cells like macrophages and neutrophils play a major role in cleaning up this cellular waste. So what activates the complement pathway? So we have some initiators. The immune complexes or antigen-antibody complexes. Apoptotic cells or dying cells, certain viruses and gram-negative bacteria, the C-reactive protein or the CRP bound to the ligands. For the alternative pathway for the initiators, we have various bacteria, fungi, viruses, and tumor cells. It does not require antibodies, and this is activated directly by the pathogen surfaces. For the mannose binding lectin pathway, some initiators are the microbes with terminal mannose groups on their surface. This is activated when the mannose binding lectin binds to those microbial sugars, which then triggers the complement activation. So the complement components circulate in the blood in an inactive form under normal conditions. Regulation is essential to prevent uncontrolled activation that could damage host tissues. For complement control proteins, so control proteins inhibit excessive or inappropriate complement inactivation. For major regulators, this includes the C1 inhibitor, which blocks the early steps of the classic pathway. Factor 1 can inactivate C3b and C4b, preventing formation of complement complexes. Factor H regulates the alternate pathway by binding the C3b and preventing overactivation. And C4 binding protein controls the classic pathway by inactivating C4b. Complement activation involves a series of enzyme activations, and each complement component is initially inactive and becomes activated by the preceding component or complex. For enzyme cascades, so complement activation follows cascade reactions where one enzyme activates the next. These cascades amplify the response, allowing a small initial trigger to produce a strong defensive effect. C3 cleavage, so C3 is a central complement protein, and it is cleaved by the activated complement enzyme cascade. The cleavage of C3 will lead to the opsonization of pathogens, recruitment of inflammatory cells, and formation of the membrane attack complex, or the MAC. Complement receptors are surface membrane glycoproteins expressed on various immune and non-immune cells. They bind fragments of complement proteins, mainly C3 fragments, produced during complement activation and degradation. For the function, so it enhances phagocytosis, so the receptors help immune cells recognize opsonized pathogens. It can facilitate immune signaling, so binding of complement fragments, which will trigger cell activation and cytokine release. and it helps with coordinate the immune responses. So it can help promote communication between innate and adaptive immunity. Some major functions of the complement system is opsonization. So opsonization is the complement protein like C3b, which coat pathogens, making them easier for phagocytes to recognize and engulf. We have recruitment of phagocytes, so complement fragments act as chemoattractants, guiding phagocytic cells like neutrophils and macrophages to the site of the infection. For cell activation, so we have binding of complement fragments to complement receptors on immune cells, which will trigger the activation and lead to cytokine release and enhanced antimicrobial activity. For the immune complex removal, so the complement will help clear antigen-antibody complexes from circulation, preventing deposition in tissues and tissue damage. For cell lysis, so we have the formation of the membrane attack complex, or that MAC, which leads to lysis of target cells, especially pathogens with membranes. So elevated complement levels, this can be caused by many inflammatory conditions like trauma and acute illness. The reason is so we have complement components such as C3 will actually act as an acute phase reactant and increase during times of inflammation or trauma or stress or things like that. For decreased complement levels, this can be caused by excessive complement activation, which leads to the consumption of those components. It can also be caused by a deficiency of a single complement component due to genetic defects. So effect of this, so it can reduce the ability to fight infections or clear immune complexes. Complement deficiencies can increase susceptibility to pyogenic or pus-forming infections. Different types of deficiencies affect specific complement functions, so it depends on what type of deficiency you have and how that would then be affected. So the types of complement deficiency, we have deficiency of the opsonic activities. This impairs opsonization, reducing phagocytosis of pathogens, and this can lead to an inefficient clearance of bacteria and higher infection risk. But then we have deficiency affecting lytic activity. So this is any deficiency that compromises the formation of that membrane attack complex, or the MAC, and it results in reduced cell lysis, particularly affecting the Neisseria species. Then we have deficiency in the mannose binding lectin pathway, and that impairs activation of the complement via the lectin pathway. It reduces early innate defense against pathogens in mannose-rich surfaces. Some disorders that can be associated with the complement deficiency. So first up is we have the rheumatic disorders with immune complexes, and certain autoimmune or rheumatic diseases can involve immune complex formation. The complement is consumed during immune complex clearance, which can contribute to tissue inflammation and damage. Next one is infectious diseases. So complement deficiencies or dysregulation can increase susceptibility to infections. Inadequate opsonization, lytic activity, or pathway activation reduces the body's ability to control microbial growth. And then the last one is deficiency of the control proteins. So control proteins normally prevent uncontrolled complement activation. This deficiency can lead to excessive inflammation, tissue injury, and an increased risk of immune-mediated diseases. Some indications for complement testing is, having a recurrent pyogenic infection. So certain individuals with complement deficiencies are going to be prone to have repeated bacterial infections. Some of these common pathogens can include the Neisseria species like meningococcal meningitis or Streptococcus pneumoniae. Deficiency in opsonization or lytic activity is often responsible for these. Then we have angioedema without urticaria. This is caused by deficiency of the C1 inhibitor, and this results in localized swelling without typical hives. And then the last one is autoimmune disorders. So complement deficiencies can contribute to autoimmune diseases. And how this works is it is impaired clearance of immune complexes in apoptotic cells, leading to immune system attack on self-tissues. Some common actions of these cytokines. So cytokines are signaling molecules secreted by immune cells. They are produced rapidly in response to cell activation. And the mechanism of action, or how they work, is they bind to specific membrane receptors on target cells. And they regulate immune cell behavior, including those T and B lymphocytes. They can also influence receptor expression, which can lead to positive amplification of immune responses and negative feedback to control or dampen responses. For target range, cytokines can act on various cell types, which are not limited to immune cells, and they can coordinate innate and adaptive immunity, inflammation, and tissue repair. For redundancy, so multiple cytokines can produce the same functional effect. This ensures that immune responses are robust even if one cytokine is absent or inhibited. For local action, cytokines often act near the site where they are produced. So autocrine action, which will act on the same cell that secreted it, and paracrine action, which acts on a neighboring cell. This allows precise and localized regulation of immune responses. Cytokine interaction, so cytokines can influence the synthesis and activity of other cytokines. This will create a network of signaling that coordinates and fine-tunes innate and adaptive immunity. Interleukins. So interleukins are signaling molecules produced by lymphocytes. They primarily act on other lymphocytes to regulate immune responses, and they do not bind antigens directly. Their function is to mediate local interactions between leukocytes to coordinate immune activity. They also work to modulate inflammation and immunity by influencing the growth of lymphoid cells, the mobility of immune cells or migration of those immune cells, and differentiation into specialized immune cells. Interferons, these are part of the body's natural defense response against microbes like viruses, tumor cells, and foreign antigens. Their actions, they have pretty broad actions. They are to enhance expression of specific genes involved in immune defense. They also work to inhibit cell proliferation, limiting pathogen spread or tumor growth, and augment immune effector cells such as NK, or those natural killer cells, and T lymphocytes. Their functional roles, so we have an antiviral agent, which will interfere with viral replication and infection. Immunomodulators, they regulate immune system activity, enhancing defense or preventing overaction. Then we have anti-eoplastic agents, and these contribute to tumor surveillance and suppression. Tumor necrosis factor. This is a cytokine in the acute inflammatory response. It is particularly important in defense against gram-negative bacteria and other infectious microbes. The function of the tumor necrosis factor is to recruit immune cells. So it'll stimulate neutrophils and monocytes to migrate to the infection site. It also will work to activate of phagocytes. So it will enhance the microcytal activity of neutrophils and monocytes, and eradication of microbes will coordinate the innate immune response to efficiently remove pathogens. Hematopoietic stimulators. So these hematopoietic stimulators are molecules that regulate the growth, differentiation, and function of blood and immune cells. They are essential for maintaining adequate immune cell populations. So the major hematopoietic stimulators, the first one is we have a stem cell factor. This supports survival, proliferation, and differentiation of hematopoietic stem cells. It binds the c-KIT receptor on progenitor cells. And then we have the colony stimulating factors. These stimulate production and differentiation of specific blood cell lineages. And then we have transforming growth factor beta. So this will modulate proliferation and differentiation of hematopoietic cells. It can inhibit or stimulate immune cell development depending on the context. Then we have the chemokines. These direct the migration of developing and mature immune cells to specific tissues. They play a role in cell trafficking during immune responses. To monitor disease activity, immune and inflammatory markers are used to track the progression of the disease. This can help determine whether a disease is stable, improving, or worsening over time. To assess the therapeutic response, so this will evaluate the effectiveness of treatment in inflammatory or immune-mediated diseases, and allows clinicians to adjust therapies based on patient response. For detection of complications, so immune and inflammatory markers can help identify complications of a known disease early. This can enable prompt intervention to prevent further morbidity of the patient. So acute phase proteins are a group of glycoproteins whose levels change in response to inflammation or tissue injury. They are part of the acute phase response, which is a systemic reaction to stress or disease. Some triggers of the acute phase protein production are inflammation, or like an autoimmune or inflammatory disease, infection, so bacterial, viral, or even fungal pathogens, malignant neoplasmas, so we have tumors, cancers, things like that, various diseases or disorders, so chronic illnesses usually, trauma, so injury or tissue damage, surgical procedures, so we have tissue injury from those, and drug responses, so certain medications can alter your acute phase protein levels as well. For rapid synthesis, this will occur after tissue injury or inflammation of these acute phase reactants. Their levels show varying degrees of change. So some proteins will increase, which will show a positive acute phase protein, and some proteins will actually decrease, so they'll have a negative acute phase protein. Some examples of these acute phase proteins are your C-reactive protein, or your CRP. This is one of the most commonly measured inflammatory markers, and levels rise rapidly during infection and inflammation. Next one is your procalcitonin, or your PCT. This is used clinically to help detect bacterial infection in sepsis. Levels increase significantly during systemic bacterial infections. And then we have alpha-1 antitrypsin, which is a protease inhibitor that protects tissue from enzyme damage during inflammation. Last one we're going to talk about is ceruloplasmin, and that is a copper-carrying plasma protein, and it plays a role in iron metabolism and oxidative processes. So different acute phase proteins rise at different times following an inflammatory or infectious stimulus. So understanding their kinetics can actually help in diagnosing and monitoring. So some response times of these, particular acute phase proteins. So we have the CRP, and that's about 6 to 10 hours. Fibrinogen is about 24 hours. Then we have your complement component C3, and that's about 48 to 72 hours. And then we have your C4, and that is also about, 48 to 72 hours. So CRP responds rapidly, which makes it a sensitive early marker. Fibrinogen rises more slowly, which can contribute to later inflammatory responses. Your complement C3 and C4 take about that 2 to 3 days to increase, which can actually reflect ongoing immune system activation.